All right, guys, here we go. Kinesiology 230, Chapter 1, yet again. This is presentation 2. We're talking about the muscular system and skeletal muscle, but more on the microscopic side. Now, we introduced this a little bit in presentation 1, but we're going to dig in a little further here in presentation 2. Let's go ahead and jump in. We saw this earlier, and we saw the epi, peri, and endo. If you remember, the epi was the EpiPen, and the epimysium is the outermost layer, and we had to compare and contrast between the muscle belly and like a bridge cable. We looked at the epimysium being the outermost layer, the epimysium being the outermost layer up here. And then you have the fasciculus itself is being held together by the endomysium, which is denoted, and then, I'm sorry, the paramysium first, sorry, and then the endomysium. And yeah, to me, it was kind of like the striking comparison between the bridge cable and our muscle. So let's go ahead and take a look at this a little bit further. So we have this kind of system in place, and within the context of the last presentation, it focused on everything to the right, that's in this kind of blocked out section here. The muscle itself, the epimysium, the fasciculus, the paramysium, and the endomysium. If we go ahead and go the next step in this equation, we get rid of that, we now look at this part. And this is a much smaller part yet again. And, this gets us down to the single muscle fiber that is within here, within the actual epimysium itself. So again, we're going from that bridge cable, if you can think of it like big picture bridge cable, all the way down to the individual strands that are in the cable itself. We're going from an overall muscle to the fasciculus, and then we're going a little bit further into the endomysium, and the endomysium, now we take it down to a single muscle fiber itself within that. So we have the sarcolemma, the sarcoplasm, which is the inside of the sarcolemma. The sarcolemma itself is like the vessel of the container. The sarcoplasm, you can think of it as like plasma inside of the container as the sarcoplasm. You have the myofibril, which is this guy here, which is a smaller yet again component of the sarcolemma. And then within that you have myofilaments, which are smaller yet again component of the sarcolemma. So you kind of see how we've taken this from big picture, from like the muscle itself, we've narrowed and narrowed and narrowed and really tightened our focus and became more microscopic with this whole process. So again, remember, sarco is flesh in Greek, a lot like we talked before about epi, peri, etc. Sometimes you have to just remember these anatomical terms. And even with that, when you look at myo means muscle, whereas sarco means flesh, right? So what you have is you have these different, if you will, groups or regions on the globe who were learning about the human body at kind of roughly the same time. Some were Greek, right? And they were coming up with, okay, that's sarco, that means flesh, whereas you had myo, muscle, was being introduced elsewhere on the planet. So let's take this back to the sarcolemma. With the sarcolemma itself, excuse me, each muscle fiber, it's encased in a plasma membrane. That's that sarcolemma. And the plasma that's in there is that sarcoplasm. And it does a couple things here. It encloses or encases the actual cell. It regulates transport material in and out of the cell, which we'll talk about at a cellular level here in a little bit. And then also you have what are called action potentials, and those are electrical impulses that are either shot down to the muscle to contract, or it's sensory and it sends a signal back up to the central nervous system to process and interpret, to then send a signal down to contract. So it's a constant communication that happens up and down the central nervous system, coming from the muscle up to the central nervous system and then back down to the muscle, which we'll dig into that a little bit further. All right, so let's go ahead and jump into the next slide here. When you look at the sarcolemma, so real quick, we've kind of taken the sarcolemma, and now we're going to go ahead, we have these bundles here, right, the sarcoplasm. And if we go ahead and look at this is a myofibril in here. The sarcolemma is this kind of white outside, and then you have the sarcoplasmic reticulum inside, that's the yellow. And the sarcoplasma, the sarcoplasma in here is that kind of membrane, if you will, the membranous type of fluid that's on the inside of the sarcolemma. So here we're looking at a much more magnified view of the sarcolemma. Within this, the sarcoplasm, is that watery solution that contains the energy source of a cell. And it's interesting how it works and what triggers that. You have the mitochondria, which is kind of this purplish blue up here. It's where the ATP is produced. And you often hear like the mitochondria is the powerhouse of the cell. And it ain't a lie. It's not a lie. It's where ATP is produced, and that's what causes the muscle contraction. The sarcoplasmic reticulum itself, that's these kind of yellowish tubes in here. And these yellowish tubes, they store calcium, and that's what regulates muscle action and allows ATP to actually bind and make a muscle contraction happen. Now, the transverse tubules, now I will say it's transverse to the sarcolemma itself, because if you look at this picture, this would not be like in a transverse plane in this rendering, but if you can just imagine taking this thing and turning it, it would be transverse. And sometimes you have to kind of look at this, and I'll get myself out of the way here, and maybe stand on your head. But adenosine triphosphate, ATP, is created in the mitochondria. That's this up here. And it's found in the sarcoplasm, right? That's that kind of fluid. And the sarcoplasmic reticulum releases calcium into the sarcoplasm of a cell when an action potential passes through the T tubules. Now, this is a T tubule here, and we'll talk a little bit more about that, but that's where you have the electrical impulse come in from the extension cord that is our central nervous system and peripheral nervous system. So with this is like, again, these are like the things you may have to wrap yourself around. You kind of have to just read these over and over again. You may have to take a notebook out and rewrite them a couple times, but this whole passage here is fantastic in terms of, well, when you look at the boundary of a sarcolemma, but outside the nuclei is the cytoplasm, referred to as a sarcoplasm. In the muscle, it's a watery solution. The watery solution contains the cell's energy sources, such as adenosine triphosphate, ATP, et cetera, et cetera, et cetera. I'm not going to belabor you to sit through me reading this, but I would take time to find this in the book and read through it, find this in the book and read through it, because these are the types of things you have to kind of commit to memory going forward in this class. So I'm going to break off from here and go to the next presentation.